All right, let's jump right in. We are going to talk about one of the most game-changing scientific tools of our lifetime, CRISPR. You know, it's this incredible technology that gives us the power to edit the very code of life itself, our DNA.
For years, the whole idea of fixing our genes was, well, clumsy. And I love this quote because it just hits the nail on the head. It felt like trying to perform delicate surgery while wearing oven mitts. CRISPR changed all of that. It was like we were finally handed a molecular scalpel, a tool with the precision we'd only ever dreamed of. I want you to think about your genome for a second. Imagine it's a book, a massive instruction manual for you that's 3 billion letters long. Now, a lot of really devastating diseases, they don't come from whole missing chapters. Nope, they come from one single, tiny typo. And that's the challenge, right? How do you find and fix one wrong letter in a book that big? That's what CRISPR's all about.
So yeah, let's really dig into that idea. Because for so many genetic conditions, things like sickle cell anemia or cystic fibrosis, it all starts with these tiny, single-letter mistakes in the DNA code. It's kind of wild to think that these huge, life-altering problems can start from something so small.
Okay, but here's the part that is just mind-blowing. CRISPR wasn't invented in a lab from scratch. It wasn't something we engineered from the ground up. It was discovered. It was out there in nature the whole time, just hiding in the microscopic world of bacteria. This isn't just a story about human smarts. It's a story about us learning from nature's own genius.
And that's exactly what it is. At its heart, CRISPR is this super sophisticated immune system that bacteria have been using for like millions of years to defend themselves from viruses. We literally borrowed a survival tool from one of the oldest forms of life on the entire planet. And the way it works is just so elegant. So a virus attacks the bacterium, right? The bacterium survives, and it actually snips out a tiny piece of that virus's DNA. It then stores that little piece in its own genome, kind of like a molecular mugshot. So the next time that same virus shows up, the bacteria's got its mugshot on file, it can instantly recognize the invader and just shred its DNA. Infection stopped. It's brilliant.
Now, this didn't all happen at once. Scientists first spotted these weird repeating DNA patterns way back in 1987. But it took 20 years, until 2007, for them to prove, hey, this is actually an immune system. But the real earthquake was in 2012. That's when researchers published a paper showing they could hijack this system and reprogram it to cut not just viral DNA, but any DNA sequence they wanted. And that changed everything.
So how exactly did we turn this ancient bacterial defense system into this incredibly powerful gene editing tool? Okay. Let's open up the molecular toolbox and see how it works.
So, the whole system really has two key parts. First, you've got the CRISPR part, which is basically a guide. Think of it like a GPS that you program with the exact address of that typo you want to fix in the DNA. Then you have Cas9. Cas9 is an enzyme, and its job is simple. It's the molecular scissors that make the cut. That's it. An address and a pair of scissors.
It all boils down to these 3 simple actions. Number 1, the guide RNA finds the target. It hones in on that exact spot. Number 2, the Cas9 scissors make the cut. And number 3, and this is the amazing part, the CRISPR system just steps back. It lets the cell's own natural repair kits do the rest of the work.
So once that cut is made, the cell has to fix it. And it basically has 2 ways of doing that. The first one, called NHEJ, is the fast and sloppy way. It just jams the 2 ends of the DNA back together. This usually messes up the gene, which is perfect if your goal is just to turn a faulty gene off. But the second path, HDR, is the precise one. If you give the cell a correct DNA template, it can use it to perfectly rewrite the sequence. So you have a choice. You can either break a gene, or you can fix it. Now, making a clean break across both strands of DNA can sometimes be a little risky. So, of course, science is already evolving. The next generation of these tools, things called base and prime editing, are even more precise. They're designed to work less like scissors and more like a pencil and eraser, allowing scientists to chemically change a single letter of DNA without making that big cut at all. So, this amazing technology is no longer just something you find in a research lab. It has made the huge leap from the bench to the bedside. This is where CRISPR stops being a tool and officially becomes a medicine.
And this was a truly historic moment. In late 2023, health regulators approved a therapy called Casgevy. It's a treatment for severe blood disorders like sickle cell disease, and it is the world's very first approved medicine based on CRISPR technology. This is it. The revolution is here.
But what does a one-time cure like this actually look like for a patient? Well, it's a serious medical journey. As you can see here, it starts with doctors collecting a patient's own blood stem cells. Those cells are then sent to a lab where they're edited with CRISPR. Then, and this is a huge step, the patient gets high-dose chemotherapy to wipe out their old bone marrow to make room for the new edited cells. Then the edited cells are infused back in. It's an intense, hospital-based procedure.
So after all that, does it work? Well, the results are just staggering. In the main trial for sickle cell patients, just look at this number. 29 out of 31 people were completely free from the excruciating pain crises that define this disease for at least a full year. For those patients, that number is absolutely life-changing.
The approval of Casgevy is really just the opening chapter. It's incredible, but it also kicks the door wide open to a whole new set of questions about what's next, both in terms of the amazing possibilities and the really important guardrails we need to build.
And probably the most important ethical line we have to talk about is this one, the difference between somatic and germline editing. Somatic editing, that's what Casgevy does, it only affects the person being treated. The changes die with them. But germline editing would mean changing reproductive cells, sperm or eggs, and those changes could be passed down through all future generations. Because of that, scientific organizations all over the world are saying, whoa, hold on, and have put really strict limits on that.
So what's the next big technical mountain to climb? Right now, we mostly have to take cells out of the body to edit them. The holy grail is what we call in vivo editing. Can we figure out how to safely deliver the CRISPR machinery directly inside the body to fix cells right where they live, in your eye, in your liver, or even in your brain?
And then, of course, there's the elephant in the room. With a price tag that can be millions of dollars for one patient, we run into a huge, critical question. How do we make sure these revolutionary cures aren't just for the super rich? Solving the problem of access and affordability is going to be just as hard as solving the science.
And that really leads to the ultimate question, doesn't it? What are the rules of the road for a technology that lets us rewrite the source code of what it means to be human? As this power grows, so does our responsibility to come together and create some smart global rules to guide how we use it. Who gets to decide what we edit and why?
When you step back and just look at the journey CRISPR has taken, it is honestly incredible. This is a story that started with a quirky bacterial immune system and has now become a real, regulated medicine that is changing people's lives. It's the perfect example of how a wild discovery gets carefully and thoughtfully turned into a technology we can trust. The Book of Life is officially open for edits, and the next chapter is being written right now.